male flowers. And then just below that is the field of female flowers. And it's, yeah, Jim was sort of studying it there because it's quite wobbly at this point. So on these flowers, I can see some of the pollen that Jim and Steve brushed on this morning. And we can also see that the male flowers have not yet opened, which is what we want to see. Typically, those male flowers are going to open about 24 hours after the flower is, or after the bloom is sort of just peaking. And we're approaching that time sometime in the next few hours. And so we wanted to get in early and be able to make sure we capture all the pollen that we can capture.
The stamens actually heat up as well, so they're heating now. So we're taking its temperature, and it's about 8 degrees warmer than the female flowers. So they're on their way to open.
They're heating up. We should see them open up tonight.
It's pretty magnificent.
We've got a couple questions here. So will the parts that we're cutting away, will they be composted?
Yes.
Yeah, absolutely they will. And I will say that a few years ago, we actually were able to collect one of our corpse flower inflorescences to make an herbarium specimen for the National Herbarium, which is at the Smithsonian Institution's Museum of Natural History. That's where the National Herbarium is. And the herbarium is basically a natural history collection of plants. So we were able to press it, dry it, and preserve it for basically hundreds and hundreds of years as a representative of this species. But this one, we already have a specimen. We're probably going to compost it. We're also hoping that our pollination efforts this morning will be fruitful, will produce fruit. So at the base of these female flowers, there we go. You can see a nice picture of the fruit there. That's from one that we successfully pollinated in 2017. And basically, every time you see a big yellow fruit, that was previously an orange ovary that was in the female flower. Let me show you. That looks like, so there's the female flowers below, the male flowers above, and that's what we've exposed here today. But that sort of pink, orangey, bulbous structure there, that is the ovary. That's the part of the female flower that's going to develop into the fruit. It has a long stalk above it. It's called a style, and that's connected to the bulbous yellow part at the top, which is a stigma, which is where the pollen lands. And then the pollen tube will grow down there to fertilize the ovules inside of the ovaries below that. All right, let's go back to our live feed here. So what Jim and Steve are doing now is they're fashioning an aluminum piece of aluminum foil, and they're going to wrap it around the spadix just below where the male flowers are. And it's got to be pretty tight up against the spadix here, because we want to make sure that we collect all of that pollen. Another question, do we have any other questions, Grace?
Susan, we've got some questions about the stink. Does it stay on your clothes, and can you smell it when you go home?
This is a fantastic question, and I experimented with that last night. No is the answer. So it really, really stinks. And late last night, it kind of started off smelling very sulfury, like a lit match, you strike a match, and then it got a little bit garlicky, a little oniony. But by the time I left, and I know in the middle of the night, it really just smelled like would you say hot diapers?
Yes.
Yeah, I like to say it's a sort of a trash truck full of diapers in Miami in July. It's really not a good smell. It smells like rotten death and poop and all kinds of bad things. So yeah, but so the spade still smells this morning. I will say I was over the bloom last night, getting a lot of the wafting of the stench on me, and could not be detected by my family when I got home. So it did not stick onto my clothing or my hair or anything. You can see, I'm going to scan up here again. We can see this spadix is really wobbling as they work to secure that aluminum foil collar around it to collect that pollen.
And about how much does a corpse flower weigh?
This particular one was between 60 and 75 pounds. I had to estimate this one. With the pandemic, we're working with less staffing, so I repotted this one myself. So I didn't get an opportunity to weigh it, but comparing to some of the ones I did in close proximity, 60, 75 pounds, somewhere in that neighborhood.
And typically what we're weighing is the underground part of the plant, which is the corum, which is a storage unit. It's a kind of a stem that grows underground, and it will swell up from having a leaf. It'll actually store a lot of the energy produced through photosynthesis in a leaf, and then it uses that stored energy to make this bloom, which doesn't photosynthesize that much. So it's not, this one is majorly taking out energy from the storage unit. You can see there's the corum right there. And the leaf is putting energy into that corum for storage, preparing it in order to be able to produce an inflorescence.
We've got another question. How many times have we done this pollination process at the Botanic Garden before?
It's our third time.
I think our third.
Yeah, this is our third time, and we had one of the two attempts that we made in 2017 was successful. The other one was not, and that has to do a lot of times to the storage of the pollen. This is very much a work in progress, figuring out the science of how we preserve this pollen. It's a tropical plant. A lot of times, tropical plants don't respond well to cryopreservation or freezing them for preserving them. But there's a pretty good technique that's been worked out now to be able to preserve pollen to make it remain viable, make it remain useful after it's been stored. But at that time in 2017, I think the technique wasn't quite as good. And fresh pollen is always better.
So Susan, why is this so big? What's the reason that, why it could do all this without being 8 feet tall?
Part of it is, and why it's so huge, I don't know. But I can say that the spadix is that size, and in part because it is emitting this odor for up to a mile away from where it's blooming to attract pollinators. So these plants are not super common. You wouldn't find, tons of them in a forest that would all be right next to each other. They're going to be a little bit more spread out than that. So it's really important for them to be able to disperse their scent to attract their pollinators, which then disperse their pollen to other blooming corpse flowers. This separation that we see in the flowers on the inflorescence, basically what it does is it allows for this plant to not self-pollinate. And it does that through a temporal separation. And what I mean by that is that the female flowers, as I said earlier, are receptive to being pollinated earlier than the male flowers in the same inflorescence will release their pollen. And that effectively makes them not be able to self-pollinate because the female flowers cease being receptive, they stop being receptive. And then when the pollen is released, it can fall over them, but they're not going to be fertilized by that pollen because they're no longer receptive to being pollinated. So it's really just getting the scent out a mile away, basically. But it could probably do that being a little smaller.
I'll add that I know some insects are capable of seeing heat at night. This last night on a thermal camera was like a big spotlight in the room, up 10, 15 degrees warmer than the ambient temperature, so it stood out like a sore thumb. So there might be that aspect to it as well.
Absolutely, and that heat has that function, and also has the function of volatilizing the chemicals, the compounds that are made by the bloom that do attract those pollinators. I gave this example in an earlier program, but basically, if you think of chopping garlic in your kitchen, you can smell that garlic; it's really strong to you, but no one in the living room can smell it. And then you put it in a frying pan, you heat it up, and the whole house smells like garlic. The same idea here, that spadix heats up, and it volatilizes those compounds, those sulfur compounds, and spreads them out again about a mile away. And that's a great image, thermal picture of a corpse flower. You can see that spadix really getting hot there. That's 85.6 degrees in that picture.
And we've got another question about, so we're talking about attracting pollinators. What kinds of pollinators or animals are attracted to this kind of stink?
Anything attracted to that scent, carrion beetles are the well-known one, but flies, any insect that is going to be attracted to that rotten smell will fly into the spathe and visit the flowers and serve pollination.
And once we have fruit and seeds, what will we do with them?
The seeds?
Yep.
Oh,
we're going to grow them.
Yeah. We're going to grow them, yeah. And part of the conservation project, we'll be able to distribute them to other gardens who don't have them or are looking to expand their genetic diversity. This will be, if we've succeeded at pollination, another 40 to 60 genetically unique individuals. So they're worth preserving and growing.
Yeah, and one really cool aspect of that is that I said that we got pollen from Chicago Botanic Garden in order to pollinate this bloom this morning, and we actually got pollen from how many different plants was it? Three different plants. So we had pollen from 3 different plants, and we're actually contributing our samples from our Amorphophallus titanum collection to Chicago Botanic Garden's efforts to look at the genetic diversity of these plants in cultivation. And as part of that project, Chicago is actually going to be able to also analyze the offspring of this pollination attempt and to be able to tell us which of those offspring came from which of its parent plants. So we could mix that pollen, even though we're doing these breeding experiments, because we can rely on the genetic analysis to tell us which pollen produced viable offspring. Yeah, and you can see that picture is showing the pollen from this morning. They poured out the pollen from the tubes, and then what did you guys do with that?
So yeah, we had paintbrushes. We just took paintbrushes and we scooped it up on our paintbrush, and then we painted each female flower, each stigma with the pollen.
Yeah, you can see that great picture of the cutout of the spade that they did this morning. They did a couple of cutouts, actually 3 cutouts? Two cutouts, to be able to pollinate all of those female flowers. You can see they're hanging on the pollen onto the stigmas, the receptive female flowers. And then I've got the bottom part of that spade that was removed. You can see those big cutouts here, and on that side, a little viewing window that was cut out. Just to give you an idea, too, of the thickness of this. Here's a piece of it that was cut out. That's the width of that. Pretty thick, and it's very spongy, so I could actually kind of squish it down with my fingers. Quite spongy. But it's pretty heavy. Yeah.
A lot of water.
And I mentioned this last night when Steve and I did a program, but you can also see that when you look at these corpse flowers in bloom, it looks like the spade is, a solid tube, basically, a big sort of trumpet-shaped tube. But actually, it's, just a leaf that's wrapped around. It's a modified leaf called a bract, and this, because if it's a specialized inflorescence, it has its own name, a spade. But it's basically just a modified leaf that is attached at the base, and then it wraps around that columnar center portion, the spadix, and then trumpets out at the top to spread pollination and spread their scent and attract pollinators.
And Susan, with our conservation efforts, are we reintroducing to natural habitats?
We are not. So yeah, we do not have any plans to reintroduce these plants into the wild. This is an endangered species, but the plants that we have are cultivated plants. They were not wild collected plants. And typically when you're doing reintroduction studies, you want to reintroduce the sort of progeny from wild plants. So we wouldn't be reintroducing these into the wild. Our work in sort of the conservation efforts that we do is to maintain the genetic diversity and the representation of this species in cultivation.
And team, what will happen once the plant produces seeds?
It'll take about 6 months for the seeds to ripen, and then we'll plant them out fresh. They unlike some of the seeds that grow in our gardens around here, they don't want to dry out. So it's not like we'll take them and dry them out to plant another time. So as soon as they're ripe, we will take them and we will plant them out in a large pot or several large pots so that they germinate, and then we'll separate them and grow them out individually.
So once that process is over with, the rest of this will be cut away, and this plant will be repotted. I'll take the form out of the pot and wash it off and get it in fresh media and get it ready for that next cycle, which will likely be a leaf 6 months from now.
Yeah, and you can see in that great life cycle illustration that we got up there, the top there is the corum. That's that underground storage structure. There's a stem. Right up there. And then basically that corum then either produces so here's here's that quorum again, and you can see in the middle there, that is actually an old leaf that has synapse, and you cut that down, and then we've exposed that quorum for pre-potting to check on its health, etc. So then that quorum is going to grow a bud, and that bud is either going to make an inflorescence or it's going to make a leaf. And so what we're showing you today is, of course, the inflorescence, but the leaf is quite enormous as well. You can see it on the left side there. There's a little person on there for scale, if you can see that. Those leaves are, quite large. They can be, 15, 20 feet tall and wide. It looks like a tree. It's got that single petiole, that single leaf stalk, but it looks very much trunk-like. But you could, what do you use to cut the leaf down when it's done moving?
It's the saw.
Yeah. Goes through pretty quick, probably.
But it's like they're pretty heavy, and it's like a series of straws if you were to cut a cross section. But nearly just full of water, so really heavy. It's like cutting a tree almost. It's almost that way.
If you sort of knock on those petioles, that trunk-like petiole, it's very much like knocking on a watermelon. It's got a lot of water, but it's also got a lot of air space in there as well, so it's kind of a hollow sound, which is quite different than if you're knocking on like a woody tree, or a palm tree, something like that.
And we've got a couple of questions related to the stink again. So, does it still smell?
Yes.
And then we see some emphatic nods. And are there any stinky flowers that folks can grow at home?
Yeah, they can grow. There's some succulents that they can grow at home, like Stapelia. It's also called a carrion flower. It's a succulent that has long leaves. They're thick and cylindrical, and they have big flowers on them that are that have a little lighter color than that spade that we cut off. But yes, they also smell like rotting flesh.
They're beautiful flowers, sort of star-shaped flowers.
They're like a big starfish. Yep, they're big, thick starfish in a light red. And those are pollinated by flies. And actually, flies will sometimes come in and lay their eggs on it to hope that the maggots will eat it, but it doesn't last that long. And then there's also some orchids. There's a lot of orchids. Bulbophyllum is a large genus of orchids that the majority of their flowers actually smell the same way, like rotting flesh. So, yeah, can you think of others?
Yeah, there's a relative of this guy here, Amorphophallus konjac. If you really enjoy this plant, is a smaller version, basically, that is hardy to zone 6 in the U.S. So, if you want to grow something like this at home, that's an option. You can plant it right outside, and it'll handle freezing temperatures here.
And that one, the common name of that one is konjac, which is the same as the specific epithet of it. And it smells, I think, worse.
It smells pretty bad.
It's pretty bad. That's more like a fishy smell compared to this one. It's more like a diaper.
But the nice thing is with the konjac, it will grow as an annual in your garden, and so the foliage will come up every year, and it'll form a little clump, and it's beautiful because it's got mottled stems that are green, gray, and purple, and they're really beautiful in a shade garden. And they bloom in the Washington area very early in the spring, so like March or early April at the latest. So they bloom early, and so and because the smell is outside, you can smell it, but it's not overwhelming.
Yeah, it's not an uncommon strategy for plants to mimic the smell of carrion or rotting flesh to attract pollinators. We like to think of flowers as always smelling really nice and pleasant, sort of perfumey, but there's quite a few really stinky ones out there.
And can you all talk a little bit about why the second court flower looks different to the one that we just dissected?
Yeah, absolutely. Yeah, we can talk about that one. It's so it's not fully blooming yet. It still has this large outer graft on it. So this is like a graft that's protecting that growing inflorescence. 3 days a week, this one's behind the other one.
This is about a week from now.
Yeah. So soon this time next week, we might have another full bloom here, but this one's just still developing, and so it's not coloring up yet. If we look at the outside of the spade here, it's green. It's starting to get a little bit of color coming in on the inside there, but we anticipate that one's going to get maroon colored like the one we just cut off as well. But it's got some growing to do, so it has quite a few of its tracks. And you can see that the spadix on it is quite different color. It's a dark purple color. And there's a lot of color variation within this species. So you see a lot of color variation in the spade and in the spadix, a lot of height differences as well. If you tune into Huntington Botanical Garden's webcam right now, they've got a bloom that's about to go sometime in the next few days probably. And their inflorescence couldn't look different than the one that we just had bloom. It's really very, very stout and very wide.
It's like a Buddha.
Yeah, it looks like a little Buddha. Yeah, it's amazing. It's a really cool shape on that one. But really different. These are all the same species. If you think about something like tulips, all the diversity that you see in tulips or daisies, that kind of thing. It's very natural for species to have form and color variation.
And actually, these are siblings, so they are from the same parents, but they are they're open pollen or they're pollinated like we've done here, so they are individuals. So just to look like how you may look similar to your brothers and sisters, one may have blonde hair and one may have dark hair. So that's you can just see in the spadix here the difference, and also this one grew much thinner and just more slender in general. So yeah, so it's just genetic differences like you might see in your own family.
We also get some differences in the leaves. With these two, I know that this particular plant here has a characteristic of within the leaflets. They're very fine, and the leaf overall looks very lacy. Whereas this one's got much thicker leaflets, and the canopy, if you will, the leaf is much denser and just looks heavier. So even with the leaves themselves, they have their own unique characteristics.
And does the color affect its function or how much heat it produces at all?
That's a great question. It's not going to produce how much heat is produced. It's not going to affect how much heat is produced by the plant. But if it was producing heat during the day, if it's in the sunlight, then the darker spadix is going to be hotter from the sunlight on it. But it doesn't impact the plant's own ability to generate heat.
And for the species in the wild, what is the surrounding environment like? And how do you mimic that in a greenhouse environment?
Well, so the environment they're from is rainforest in Sumatra. It's very hot and humid. And in Sumatra, it's actually it's cloudy most of the year. So hot and humid, but less direct sun. We don't necessarily have to mimic that environment. It's more important to learn what the plant is adapted to. This particular plant requires high heat and humidity to produce just the size alone of the structures. And then also when it's in its vegetative state, the leaves have no adaptation or very little adaptation to direct light. So it's important to provide shade for them. So in order to get that heat, we need to shade out the sun, and then we need to provide humidity so that the leaves can handle the heat that they are seeing. So it's more important to understand what the plants are adapted to, not necessarily exactly the conditions they live in. So we are close in the greenhouse to what Sumatra would be like, but not quite as miserable. It's not quite that necessary.
Can you eat any part of the corpse flower?
No, you can.
Well, the bulb.
Yeah, you can eat the.
So actually, as we were talking about Amorphophallus konjac earlier, which is the relative that we said you can grow in the gardens, that's edible, and actually it is a staple in Asia. I ate it when I was in Japan 2 years ago for lunch one day. So it's the same corm but much smaller, and it was just cut into like 1-inch chunks and sautéed with garlic and butter. So anything with garlic and butter is delicious. But yeah, it was kind of flavor, like a potato, not a whole lot of flavor, just starchy. But I can't believe this, would kill you, but it probably wouldn't hurt you. But I don't think a lot of people eat this tuber.
Yeah, and one thing to
this
keep
form.
one thing to keep in mind with this whole family, it's the aeroid family. Despite the fact that it kind of looks like cabbage, as you can see there, you never want to eat this plant raw. So when the plants that are eaten that are in this family, they are cooked. They need to be cooked properly to basically break down some crystals that occur throughout the plant that can cause lots of injuries to your sort of soft parts in your mouth. Yeah, and you can show this little section of the spade here. You can really see some of that amazing texture. The surface of it is really kind of velvety. It's kind of a little bit waxy. It's what's called glaucus botanically. So it's got kind of a white waxy covering on the inside. And you can see the color is lighter at the base of the spade, and it gets quite dark in this particular individual. And that little fringe, if we had not removed this spade, it probably would have closed back up. Although this one was really open, so I'm not sure if this one would have done. It might have just fallen back. But typically, they do close back up and kind of protect the developing fruits inside there until the spade fully synapses and falls away.
This one started to try to close, but yeah, it unfurled so much that structurally it wasn't quite able to do what we will typically see. So it did attempt to, starting around 5:36 this morning, but it only got so far.
Yeah, and you can see we're indicating there that open spade. It really, really got broad and was quite flat. And by tonight, it was kind of hanging back low in the backside, probably unable to close all the way back up. That's pretty sad.
And are there any pests that we deal with in a greenhouse that eat the leaves or any other part of the corpse flower?
I haven't experienced any pests on the leaves themselves, but this species, even some of the common greenhouse pests, are not a big deal with this plant. The things we deal with regularly are different types of rot on the corm, fungal and bacterial rots in the corm, especially when they're in between life stages. Those are the biggest challenges in terms of pests. But insect and leaf disease wise, really not much pressure on these. They're such an exotic plant to the area, so not much interest, I guess you'd say, on the pest side.
I think you've got another question. In the wild, do these plants synchronize their blooms?
So yeah, the plants, I mean, not that we know of, right? We don't know that these plants are communicating with each other in the wild, but what you would expect to see in a population, and this is true for really most plants, not all plants, but most plants, what you're going to see in a population of the same species or in adjacent populations is that they're going to bloom within a certain period of time. So over, a month, 2 months, sometimes 3 months, you would expect to have, the same species coming into bloom and going out of bloom. And what that allows for is, again, that cross-pollination to maintain the genetic diversity of those species. And this will be a similar thing to that. So you would expect to find, several blooms in a large area that might be up at the same time or around the same time. And that allows those pollinators that are coming to visit the female flowers when the bloom is first emitting its stink, they're going to come in there. They may have already have pollen on them from another bloom that they visited. And then they will be taking the pollen from this bloom on to the next.
Got it. And back to Steve. Have we ever lost any corpse flowers because of rot?
Not yet. Not yet. We've had some challenges, but haven't lost one yet. I haven't. It happens. There's a period in the winter where it's really difficult. The length of day is so short, and it can get so cold that the plant, these plants are always trying to do something, even when they're in their dormant state. They're still active, and when the days are really short and it's cold, they really struggle, and that's an opportunity for rot to set in. So it's really important to control the water at that point and keep the plant as warm as possible. That's the challenge. Knock on wood, I haven't lost one just yet.
Basic, Jim will help me here. I think I'm going to try to move the camera a little closer to this to get a better view of it. So grab that.
Yep.
Get this.
Yeah. Oh, okay. Just
There
this?
you go. Yep. Sorry, a little jumbly here. But we can see that is this. That's the bottom of the spadix there, the top part, the sort of bottom of the top part, and then that is the field of male flowers. You can see that aluminum foil boat that we made in there to catch them, and then that is the field of the female flowers. And there's still some flies flying around in here. As Jim said earlier, those male flowers are going to heat back up when they're ready to release their pollen, and that's going to attract those pollinators back to them. A little closer view of what's going on there today.
And can you all reiterate for us what happens next in terms of the fruit and the pollen and things like that?
Well, we'll know in a few weeks whether our pollination that we did this morning took. Excuse me, if it didn't, then those will all fall off. Otherwise, in a couple of weeks, they'll start to swell and mature, and it takes about 6 months for those fruit to develop and those seeds to develop so that we can plant them back out. So nothing on this plant is fast except the quick bloom. Everything else is slow to grow, it's slow to mature, it's slow to get to flower. Then the one thing it does fast is open and pollinate, and the flower goes. But everything else is slow, so it'll be about 6 months till we can see when we have seeds to plant.
And you can see here's a picture again of our fruit from 2017. And what you can see on there is the ovaries again all swollen up, and then those little dark little sort of circles on the tips of them. That's the scar that's left from the style where it's attached. You can see on this picture a few little styles that are shriveled up and still remain there. As Jim said, we'll be able to tell fruit set sometime in the next couple of weeks. We won't get those fruits for a while, but we'll be able to tell if the ovaries start to swell up on their way to being fruits.
And does removing the spathes affect the fruit production at all?
Not at all. Yeah, it actually, it helps it to not rot a little bit. So, because what would happen to that spathe, as I said earlier, eventually this spathe, which is sort of like a glorified piece of, cabbage, it's just a big leaf here, this is going to rot pretty quickly. And so all that rot around the lower part of the inflorescence can actually cause some issues. So even if we hadn't cut it off now to be able to collect this pollen from the male flowers, we would have cut it off in the next few days to remove it from potentially rotting the bottom. And we will also, we're going to put the rest of this thing.
Yeah, at some point this spadix will collapse as well, and we'll cut that out for the same reason. It'll begin to rot. The plant will discard it naturally if you do nothing. And I've seen with other species, it'll actually develop mold on the inside, and you'll lose fruit that way. So, if anything, we're helping by removing it at this point.
Will we use the pollen we collect from this corpse flower to pollinate the second corpse flower that we have behind you?
No, because, they're siblings. So what we're going to do, we're going to send our pollen to Chicago Botanic Garden for preservation, and also to Huntington Botanical Garden if there is enough time to get it to them before theirs blooms. So to ensure cross-pollination. And then this one, we are going to hopefully get pollen from Huntington Botanical Gardens to pollinate that one when it blooms.
And whenever we have one going to bloom, we're always interested in finding out what other gardens have a potential bloom as well. So we really reach out to the network of gardens that frequently do have these come into bloom, or that we know have large collections, so that we can do this very intentional swapping of pollen. And again, it's really it's a breeding program for these plants to maintain that genetic diversity.
And will this plant bloom again? And do corpse flowers bloom only once?
That's for me is very exciting. It should bloom again. It's certainly capable of blooming again. And the question is when. And that, for me, that's the fun part. It should produce a leaf in its next cycle. So again, about 6 months from now, after the fruit is ripened, we should see it go back into a vegetative state. And then after that is when the unknown is. After that leaf is through its cycle and it senesces, it could very well bloom again, and that would be a couple of years from now. So, and then how many times they bloom within a lifetime? I know there are many in cultivation who have bloomed up to 4 times, but that's not necessarily a limit. So that's a question that hasn't been answered yet.
And how many corpse flowers do we have in our collections total?
Around
57.
60,
50, yeah. Yeah, we have a lot from the last pollination. And we have about 50 seedlings from the last pollination that we will distribute to other gardens.
Yeah, we have about 15 mature plants. Is that yeah? So about 15 mature plants that are potentially capable of blooming, and then a lot of these seedlings that we've grown up. And at the time when we had that fruit set in 2017, there was a lot of discussion about should we share out those fruits, those seeds, or should we share out plants that we're able to propagate from them? And we ultimately decided it would be better for us to grow them up a little bit, and then to be able to hand off, a healthy individual onto these other gardens in universities.
Yeah, and I will say that people come to the conservatory here and they want to see one. We bring these up when they flower so that people can see the spectacle of it. But most of the collection is grown at our production facility where we can Steve can control the environment much more succinctly. However, last year he did plant 2 in our tropics. So we do have 2 Amorphophallus titanums planted in our tropics. So when people can come back and walk through and see our collections again and see our tropics, look for those plants in the big house behind us.
Thank you all so much for answering all of our questions today. This has been truly fascinating. We've reached the end of our program, so if you all have any last words, we just want to say thank you so much, and we're getting lots of thanks from our online visitors today.